Thank you, Victoria, for this great introduction. I'm impressed that you can remember all these awards and experience. Yeah, one thing I want to add is I started at the University of Texas at Austin, and that's basically this 3D printing technology was invented there. And then I worked for the National Science Foundation as a program manager. I was managing $18 million program within NSF. Then I moved up here to help establishing this nano engineering department. So, and I have been enjoying very much this experience. So the talk today is 3D bioprinting of scaffolds and the microphysiological systems. So this is a kind of weird word, but if you think this as a tissue on a chip, organ on a chip, or human trial or animal trial in a dish, that kind of concept. So, so for this talk, I will first give a brief introduction about our motivation, our research, including why do we need 3D human tissues, what are the material and the printing technologies that are available for making that kind of human tissue. Then I will show you the capability of this kind of 3D printing technology for creating this physical, chemical, biological cues or environment to help cell to grow into functional tissue. Then I'll summarize my talk at the end. First motivation was this chart. So as people getting aging, or sometimes they have a disease, and so it'll be nice to have a technology that can repair, regenerate these diseased, damaged tissue or organs from inside to outside, from bottom to top, including skins, bones. Of course, we are focused more on soft tissue because those are more difficult to create, and they're more complex in biological phenotypes. The second motivation is actually for drug screening. This chart shows a typical process for drug development from discovery, preclinical trial, clinical trial, and then manufacturing. With a number of compounds here, and all the way down to the successful drug. And in average, it takes 12 years and $1.8 billion, $1 billion to get one drug to use in a clinical case. Okay, the main reason for that high cost and long time development is because many of these compounds failed at different stages. Okay, so that could be very catastrophic if this drug failed at a clinical trial stage. So you already spent like 7 years and about $1 billion on this. Okay, and this can be very, very bad for small companies, also a significant burden for big pharmaceutical companies. And from scientific point of view, these animal models and also two-dimensional human cell on the dish really don't represent the phenotypes. Okay, so it's very important to create so-called these three-dimensional precision human tissue models for that kind of early drug screening. Means if I can create these human, not just animal, and day one, so they can test these compounds in a much faster way. So it will save them time, also good for the patient as well. You know whether this compound will work or not, whether this compound is toxic or not. That toxic can be to this specific organ that you're testing, or can be side effects, right? If you are developing cancer drugs, if it cure cancer, but it's damaging the heart, then this drug is not functional. So if we can have this kind of 3D human tissue model for early testing, that will be a huge game change for the pharmaceutical companies. This chart shows a typical process for creating this kind of 3D human tissue, you start with imaging, right? You cannot just create random structure because heart, liver, brain all have specific topological, physical, biological shapes and functions, right? Then you design it using engineering rules. Then you find what material to help this. For instance, if you want to create a heart tissue, you cannot use ceramic materials to support these cells. You have to have some sort of soft, biocompatible material that can work with heart cells, cardiac cells. Then you do the printing part, in this case, to create this biomimetic tissue. Then you go do the testing and also applications. So traditionally, there are two types of printing technology. One is to use an inkjet printer, just like you print, patterns or words in your computer. You can extend that to three-dimensional fashion if you keep printing from bottom layer to the top. Okay, people have invented this kind of inkjet printers for that. The second is so-called micro-extrusion-based printing process by pushing out these inks or solution and form structures like that. So you can these are very well-developed technology in the field for the last 10 years, and they are low cost. But the problem, you can see that usually they are very slow. If you do this one drop at a time, for a sizable structure like this one may take 10 hours. It can be useful for printing non-biological material, but if you want to print live tissues, that kind of time scale is too long because cells don't survive outside the cultural media environment for 8 hours or more. And often these structures like this printing head, or in this case the nozzle, can damage the cells involved in this process. So in my own lab, we started this many years ago, about 20 years ago now, in terms of using 3D printing to create soft scaffold and tissues. So when we started this, Texas, UT Austin was a pioneering school in terms of 3D printing, or they call it at that time freeform fabrication. And I look at this work. I figure usually these processes are not so fast or high throughput compared to computer chip manufacturing, and but they are very good for customized product or devices. So I figure if you want to sell this product, who wants to buy it? And with that question, I figure only the patient can, is willing to pay the kind of personalized high-cost product. So we very much, at day one, we very much focus on soft material, biocompatible material that you can use to create these scaffolds and tissue models. And we have been working on that for many times with continuous support from different agencies. So I'll show you this video here, see how quick we can print. So this is a CT guided printing with images from CT scan of this vasculature network. And you can create these very complex structures in a matter of 10 seconds. So as I mentioned, that sizable printing sizable device like this will take 8 hours. In our case, we have this rapid printing process, that only takes a couple of seconds. And that's very beneficial to cells because they don't experience rough environment outside the culture media. If you want to, show where this printed product look like, so I can show you another video here that when you can pull out this printing part from the solution. So the light comes from the bottom. I have some solution here in this container. You can see once it's printed, you can pull it out, right? In the process we call polymerization. So you can do this layer by layer, but in a continuous fashion when you pull out this gelled product. So in this case is a branched tubes. We use that kind of tube to study vasculature function, or use this kind of tube for nerve regeneration, nerve tubes. So it takes a couple of minutes, still much faster than commercial ones that using nozzle or extrusion-based process or inkjet-based process. And the process actually involves many different materials, including this monomer, and you can put cells in the solution because it's a solution, and you can put some other materials like growth factors, controlled release particle drugs in the system to help the cell to grow. So you have a mixture of this solution, then you shine the light, and this light is a magic that can cross-link these monomer chains. When you cross-link them, they become polymer. And once they are polymerized, then they lock out these cells or particles in the light spot. So by that, you can hold these cells in the gel. It's very hard to tell where the cell goes, but you can tell where to polymerize the solution of these monomer chains. So it's very easy to do that. If we actually start with the PowerPoint PowerPoint software when we didn't have good support from computer science at that time, and it just if this is a PowerPoint slides with a light, in this case it's a white color, you cross-link, you make the pattern. Without light, it's still dark. Then there's no cross-linking, it's still liquid, you can wash them out, right? And so by just as simple as that, you can create different structures. So I show you a little bit difference between commercial ones in order to form this type of structures. And in our case, there's really no drop by drop or layer by layer processing, in terms of fabrication process. And you can make very smooth curvatures because you are printing this in a continuous fashion without stopping, right? Without scanning. So it's kind of several thousand faster than a traditional printer. So time is very important, and also resolution is very, very important. We can print as small as micron and even nanometer scale structures. You can put different chemistry in the solution during printing. So in this case, we actually print this so-called artificial microfish by adding different functional nanoparticles in the fish body. So in the tail, we put this so-called platinum nanoparticles. In the head, we put iron oxide nanoparticle because this is magnetic. So, and then we can print this quickly. And let me show you, this fish actually can move by itself by this so-called reaction and bubble generation. It can push the fish move forward. This is about 100 microns, so the size as small as a human hair diameter. And if you have magnetic particle in the head, you can see you can use external magnet device to guide the fish to move, right? It's very cool. So we got lots of press report by Washington Post and Wall Street Journal. And then in the fish body, we can also put other functional nanoparticles, in this case called a detoxification nanoparticle, that can remove toxin in the blood. So we show that once these toxins are removed by the fish, this fish turns from transparent body into colored, in this case, a red color. So you can see we have this beautiful red color fish. And the Wall Street Journal has this nice picture, imaginative picture showing this kind of micro-robotics or nanorobotics getting into people's body to remove toxins, to do sensing or to do small-scale surgery in local place, and also deliver drugs if that's the necessary part of the work. But we didn't do that. It's really the imagination of the reporters. But certainly this gives possibility in the future that this kind of nanorobotics could be manufactured, or in this case 3D printed with functions. Okay, and the idea wasn't new. Actually, back in 1966, there was a Hollywood film called a Fantastic Voyage that you can invent this kind of nanomachine, get into the blood to remove the blood clot in the patient. In this case, it was a very funny movie if you would like to watch it. Then I would like to show that we can also implement mechanical function of the printed device. So usually, in this case, I would like to talk about this so-called Poisson ratio. Means when you stretch materials, usually it gets smaller, smaller, and then break. So that's, I mean, almost 100% of the material behave like that. But we can actually print the material when you stretch it, it actually expands. Okay, so it's against nature, but there's possible new application of this kind of engineered functional materials. So this is a video shows that this kind of we call auxiliary pipe. Usually when you stretch it, this tube will shrink. But as you can see from this video here, this tube didn't shrink. It actually expands a little bit. And as an engineer, we are not limited to by this kind of design. We can make so-called hybrid structures. In this case, you have one side shrinking when you stretch it, the other side actually expands. So imagine you have new type of Band-Aid or other filling material that you need for surgery, and this thing can actually hold the space when it's inside and won't create gaps between the implant and the host tissue, for instance. And we do have this work patent by U.S. office. So if you're interested, feel free to talk to Victoria for licensing of this, very novel idea. And last, I would like to show you that you can actually put live cells in the gel before you print. So in that case, you can create live tissue instead of just a piece of plastic. Usually people do in terms of 3D printing. So one work we did was this so-called human liver tissue models. Okay, if you go to the supermarket, you get a liver tissue. In the case of pig or others, you can cut it open and look at under microscope. That's the micro. You can see the structure, microarchitecture with hepatic cells in these hexagon patterns, supported by these lines of endothelial cells and other supporting cells. Okay, and in the middle is the central vein that help to deliver the nutrition. So it looks simple, but the liver actually is a very elegant, highly functional, metabolically and energetic organs. So there are companies doing this kind of printing of liver tissue, but usually they don't have the ability to resolve the microarchitecture like this one. They don't have the ability to put different cell types in an organized fashion, and they don't necessarily have the right supporting material to help the cell to grow. Okay, so we actually developed this process we call two-step printing process. First step is to print the hepatic part, the liver part. So we developed this material called gelatin methacrylate. It's a kind of denatured hydrogen, collagen that you often see in people's body. Okay, you can use that and mix with the cells. In this case, we call it human-induced pluripotent stem cells. It means that we take the patient's skin cell and reprogram them into functional cells like liver cells, nerve cells, or heart cells. In this case, it's liver cells. So it's not embryonic stem cell. It's your own skin cell. You can program them into functional cells. So we mix these cells with the hydrogel solution here, and then put it in this printer and print. In a matter of a couple of seconds, you can form this sizable liver patterns. Okay, so in this case, I have the green. We actually stain the liver cells so you can see them under the microscope, and we intentionally make these areas, dark areas are open, so we can put another types of cell to support the liver part. So in this case, I have human endothelial cells and also adipose-derived stem cells. These are fat cells. And mixed with gelatin plus hyaluronic acid, that's another actual cellular matrix material that's in the body. And once you mix them together, you print them with this kind of pattern that you left over in the last print. So in this case, I have the endothelial cells mixed with this new hydrogel and then form these patterns. So if you look the overall structure, you have this kind of very nicely organized human liver model. So hepatic cells in the middle, and supporting cells in red surrounding these hepatic cells in a very organized fashion. And this is 3D reconstruction, you can see that, and it was published 2 years ago. Then we want to make sure it's a liver, right? Because these are skin cell derived liver model. So you measure the genes, so they make sure they are very good liver tissue. So we compare our 3D model, we call triculture model, means I have liver cells supported by functional endothelial cells and adipose-derived stem cells, and compare that with traditional 2D human cell on the dish, like many people do in the industry now, or 3D, but without supporting cell, only liver cells, okay? So you can see our model, actually stand out much better in terms of these gene analysis, okay? And then we also make sure they do have the liver function, especially for albumin secretion and also urine production. Again, we compare our 3D model with the simple 3D model or 2D Petri dish model. And you can see over the period of 20 days, we actually can culture them for a long time after printing. So we can keep this liver cell in the dish for many, many days and analyze their function. So you can see our 3D model actually, again, function better than the 2D dish and also 3D like liver cell only models. So it's very important to have liver cell with support of these additional cell types. And then last, we want to make sure this kind of 3D printed liver tissue can be useful for testing drugs. As I mentioned, that pharmaceutical companies are looking for this kind of in vitro tissue model, especially human tissue models, right? So we actually test one antibiotics called, in this case, and we put the antibiotics, this is known antibiotics, in the dish, in the 3D printed tissue culture, and analyze these enzyme expression levels. And again, the 3D, so-called trial culture model in our case is much better compared to other traditional tissue models. So hopefully, this kind of human liver model can be useful to test these unknown drugs that the pharmaceutical companies are trying to develop. And you can test that in day one after they figure out the compounds, instead of waiting for 10 or 20 years to know whether this drug is toxic, whether this drug is effective or not. Okay, so it's very, very nice model for them to try. And also, these are in vitro models, so they can quickly look at them, how these cells are performing, instead of recruiting human patients that you know that that kind of study takes time, takes a lot of money to do that, and often we don't have that good control about the human patient because they can have a lot of alcohol drinks and then will mess up your data. But in our case, these are engineered human tissue. We can control them in a very nice fashion to show good, consistent results. So that's basically the liver part. I also would like to show you that we can print this human heart tissue that can beat, just like this one. And you can see, these tissue are hanging over these two so-called cantilevers. And when the heart beats, they can bend the cantilever, and that data can tell, of course, you can measure the beating frequency, and also you can test how forceful this beating is. Now, if you have a drug, want to test whether this drug can speed up the beating frequency, or whether this drug can reduce the force or enhance force, you can do all these studies with this kind of model. So this is ongoing. And in the future, people are talking about human on a chip, right? You can print liver, you can print heart, you can print gut, you can print many other things. And you can organize these miniaturized microphysiological tissue or organs, to to have a systematic study of the drugs or other disease mechanism, right? For printing, you can create a disease model instead of being just healthy models. I can change the environment, make these cells sick, and then test the drug whether this drug can cure these disease cells or not. So there's a lot of promising promising applications with this kind of 3D printed human tissue or tissue model, in this case, human models. So to summarize my talk, I have shown you that this light-based processing in terms of 3D printing is very fast, and it gives you cellular scale printing resolution, which is very important because these cells are on the order of a couple of microns. And traditional printer cannot resolve that kind of resolution. And you can build the tissue with good control in terms of mechanical, chemical, and biological functions. Okay, and then we have many different types of material to support the tissue growth. Okay, tissue growth, and the work has been carried out by generations of talented students. These are when we were in Texas and here in San Diego, and we were heavily funded by many government agencies. At this moment, I have 3 NIH grants. Usually, for engineering faculty, this is quite significant. If you can get 1 or 2, that's already great. And we're lucky to have many of them. And we were also funded by the CERN, California Institute for Regenerative Medicine. So thank you all for voting that proposal in California. I guess it's 71, Proposition 71. And we are also funded by Congressional Funded Medical Research Program in terms of Department of Defense, National Science Foundation. And with all these fundings over the years, we thought the technology has been matured enough. It was time to create a company, and so that's why I'm here today to by Victoria to show this company called Allegro 3D. That means you can fast print things, and the company's developing these 3D printers and bioprinters, and then they can also offer you the materials for printing. Not every material can be printed. Right? And in case you don't want buy a printer, and the company also offer printing services, like send you these scaffolds devices or bioreactors for cell culture and research, and they can also sell you these 3D printed tissue models, including liver. I have shown you already, heart, nerves, cornea, retina, and the cancer tissue. So we actually have been doing many of this work in over the years, and the company are taking over the major intellectual properties for that. So with that, I would like to thank you for your attention. I would be happy to answer any question you may have. Thank you.
So in hepatic liver studies, I mean, the goal was to, if you have a artificial liver, that you can look at toxicity of various drugs. So I see that you show data that you say your cells are viable up to 20 days. And so what is your readout? So if they're viable up to 20 days and someone would be interested in testing a drug, so they'd add the drug to the matrix, and then what would be the readouts that would indicate whether the drug is toxic or not? You gave some of the enzymes of the liver and whatnot, but what do you foresee as being kind of the readout for the drugs to say if they're very toxic or not toxic?
Yeah, so as you can see, that paper was recently published, and we are actually working on the topics like you just mentioned, and those are not published yet. So I I will keep it for a while until it's published. But it's very important. These cells, we can culture them for a long time. And 20 days in that time is the goal we set. But for many of these tissues, like heart, we can culture them for a very long time. A year, for instance, if we have to do. But usually for drug testing, you do want to test quickly within a couple of days, 5 days, 10 days, and see the outcome. So in the case of heart, you want to see heart beating, you want to see the calcium signal, you want to see the fourth generation. For the liver, there's also many other physiological readouts you want to look at.
From my understanding, some of the advantages of certain companies that have a better hepatic model, it shows it can be viable much longer because you have to look at toxicity over a period of the longer the better to see actually the toxicity of the compound.
Sure, yeah. So as I mentioned, for that particular study, we just cut off by 20 days, but these cells are still alive. You can keep doing that. So I think at that moment, for that specific work we published, I think we were not looking at that long-term toxicity data. So, but definitely those are the things that we as a lab we want to continue to investigate, and with additional funding. So a lot of time we couldn't do more because we don't have enough money. Yeah, that's a different grants. Yeah.
What about density of cells?
So
if the cells get too dense, then they'll go into apoptosis and whatnot. How do you prevent that over a period of time?
Yeah, so that's a very good question. So at this moment, we put a couple of million cells per meal, and they don't overflow the culture. But definitely, if you culture them for a much longer time, they do. And I think you can use certain drugs to prevent them from growing too fast. We did that for heart cells. Yeah.
Professor, do you see any possibility of using 3D printing to create organ replacements where you don't need drugs to suppress the immune system? Thank you.
For organ, right? Yes. Yeah, so this is the holy grail of the field in terms of 3D bioprinting. Most of the time, these organs are so complex, very sizable, like whole liver, whole kidney, whole heart. I don't know we are ready for that yet. No one knows where this is going at this moment. I think it takes very, very long time to get organ scale work. But you never know when with continued funding from the government agencies, with funding from private industries, someday we will. But at this moment, it's very, very hard to make complex tissue organs with multiple cell types. For instance, in the case of heart, you have nerve support, you have cardiac cells, where you have many other cell types, you have blood supplies, and it's very challenging. Whether these immune safe, immune compatible, I think that's another important work people are working on now in the field. So that rely on what cell types you put, right? So assume if you use the patient's own cells, that is better. If you also use the patient's body tissue, we can actually decellulize and take those materials to print. That's better. But very often, this moment we use synthetic materials like a gelatin or polyethylene glycol sometimes. So definitely, there's possible issues of immune rejection. But again, this is part of the research work in the field to improve that part of in terms of immune rejection.
Have you done any work with cartilage?
We did something with Professor Daryl DeLima over the scripts before, but he's a major guy for that. So I try to stay away from hard tissue like bones and focus more on the soft side. And because on the other hand, I think soft tissues are more challenging to make because the materials are so soft, and also more exciting to work with as well because there's not so much work being done in the field yet. And for bone cartilage, I think that's the most well-developed field in bioengineering or tissue engineering, and I don't want to get into that crowd if it's possible. But definitely you can, put a mesenchymal stem cells with those gels and try to create bone models or cartilage models for disease studies or for regenerative medicine purpose. I didn't do that much, so I focus on the soft tissues.
I have a question. So obviously the cost of doing human clinical trials is very high, as you pointed out in one of your slides. What can you estimate the cost is for running some high throughput screening or even low throughput screening on these biomaterials for drug toxicity or drug resistance or drug activity?
That's a very good question because when we talk to pharmaceutical people, they ask, I want a high throughput, okay, 96 well plate or even bigger. And I think that's the main problem with traditional 3D printers. When you do it, one nozzle at a time or one inkjet time or within the glass slides or other really not industrial scale platform, it's very hard to sell them. So that's why I think many companies doing 3D printers or bioprinters having trouble with that. So in our case, that's different. I just send a light to the areas so we can easily scale up for high throughput printing. And again, for each well, it's very, very quick, a couple of seconds. So 96, you can do it within a couple of minutes. I think that's what the company is trying to push forward as well. Yeah.
Thank you.
Thank you. I have a question about, you just talked about two areas. One is the application in drug screening, the other is the biomaterials. Which area do you think will be accepted by the pharmaceutical industry or just general industry faster?
For pharmaceuticals, they want a tissue, right, in high throughput fashion. So they don't care what type of biomaterial you use as long as it's supporting the cell growth. So I think the tissue is the most important part for them.
Other questions?
There's another one there.
Over. Oh, sorry. Excuse me.
Sure.
Yes, you do.
Yeah, I will talk way now. Hi. My question is, where is your lab facility? May we make an appointment, come in and observe what you do?
Sure. So our research lab is on the UCD campus, structural material engineering building. I'll be very happy to show you what I can show. There's something I cannot show because of the intellectual property protection.
I would recommend you make an appointment.
Yeah. So
Call and find out when he's available or email.
Yeah, I will give you a card. So there's the email and the phone.
You are 825 or 924. You are out. Thank you very much.
I'm 7-Eleven.
7-Eleven. Wow.
Do we have any other questions? Yes, no? Well, if not, certainly, you know, we can speak or ask questions in the break time, the networking time, and I would encourage you to do that. Please don't ask him questions here, because then he won't get anything to eat, and we do want him to have something to eat, as we want you to have something to eat. So with that, I would like to thank you for coming. Thank you for providing this lovely seminar and your expertise and telling us all about your new company, which is very exciting. And to all of you, thank you so much for coming. Please come back, look at our website at innovation.ucsd.edu for all the various programs that we have that are available to the community, to faculty, to staff, to students. Take advantage of everything that we offer, or pick and choose. But we're happy to do this for you, and we hope it will bring you some insight into not only intellectual property, but the work that goes on at UCSD, the work that goes on that our professors are doing. And we would love to get some feedback from you. If you have any suggestions or comments, please come and see me. So with that, thank you so much. Please thank our speaker.
Thank you.